All right, everybody. So, what we're after today is looking at linked versus non-linked traits. And we're going to go over a AP type of question. And we're going to tie this because the AP question really ties back into chromosomes and meiosis. So, that's what we're going to do a little bit. So, if we're talking about autosomal non-linked traits, first of all, we need to know what an autosome is. And an autosome, especially when we're looking at like the karyotype of a human, we have 22 autosomes. And we're diploid, which means that for the autosome number 22, we have one backup copy of all of those genes. And we also have a set of sex chromosomes. If you are female, you are XX. If you're male, you're XY. But we're gonna get into sex-linked traits, but for now we're looking at autosomal linked versus autosomal non-linked. And basically the non-linked thing is a classic Mendelian Punnett square kind of stuff, where if you're talking about um, different traits, they're on different autosomes. So if we're talking, and again, I know this is like human karyotype, but if we're talking about like plant height, that would be on a different chromosome than pod shape. We have different traits on different chromosomes. And you get, when we talk about homologous chromosomes, again, that's talking about the diploid nature of this. You get backups, you get one from mom, one from dad. And again, the same genes, the same codes for the proteins exist on both. And they're going to be in the same order. We might have slightly different DNA sequences, and that's where we end up with alleles, because the proteins that are made can be slightly different from one another, which could ultimately affect the traits. So, Mendel, and this is going to be important because we're going to get into this P1, F1, F2, but Mendel was really lucky because he's looking at 7 different traits and none of them were linked. They were all on separate autosomes, separate chromosomes. So, like that is incredibly fortunate. And when we start looking at this P1 cross is all about purebreds or true breeding. So if you take this round and pollinate it with another round, they're always round, always round, always round. So they're true breeding. And that's what we would call then homozygous. And we take the wrinkled ones and we breed them just to wrinkle, just to wrinkle, just to wrinkle, and we always only get wrinkled seed texture. That would be homozygous. Now, when we cross the P generation, we end up with heterozygous F1 generations. And then we know that these alleles or these genes are distinct elements because it's not just blending, but as we take this heterozygous, the next generation, if you took a hetero to a hetero, we end up getting some of these alleles back, some of these phenotypes back in the next generation, which brings up this idea of dominant and recessive. So if we're going to define dominant, it's going to be what allele is going to show up in the phenotype of the F1 heterozygous generation. And the recessive allele would not show up, and it's only able to be seen if it is in a purebred form. So, on to the setup for this question. And with all this Punnett stuff, we get into representing alleles with letters and the dominant and recessive. When we talk dominant, we're talking capital letters. We're talking recessive, we're talking lowercase letters. So we're talking in a certain species of plant, diploid number of chromosome is 4. So basically we're talking about 2 autosomes and it's diploid, therefore there are 4 chromosomes. Flower color is controlled by a single gene in which the green allele is dominant to the purple. So again, we have this trait, which would be flower color. We've got dominant and we've got recessive, and we're going to use those letters to represent it. Plant height is controlled by a different gene in which the dwarf is dominant to the tall. Individuals of the parental generation with the genotypes GGDD and GGDD were crossed to produce F1 progeny. So again, that's what it's giving you. It just says F1 progeny in the prompt. It doesn't tell you what it actually is letter-wise. So you need to understand, if you take a P generation that's GGDD, cross it with another true breeding recessive allele, um, a purple tall plant, it would be like this, and when we cross these up, we end up with this F1 generation. So I'm going to take you through that now. So as we go here, again, we're talking about a dihybrid P1 cross, and then the P1 cross, we're talking the parental generation. That they're purebreds, they're true breeding. And again, keep these letters in mind. The flower color and the plant height are the traits, and the letters represent the genotypes, how it's arranged. So our P1, we've got a capital GG, capital DD. So now we need to picture what that means, because we want to tie this back into meiosis. So when we're talking diploid, what we're talking about is having a backup set of the same autosome. And in this case, the trait for flower color is green on both sets of those autosomes. When we talk about the traits being non-linked, flower color and plant height being non-linked, what that means is that we're talking about them existing on a separate autosome. Now, if we think about the other P1 here, we got little g, little g, little d, little d. And again, this whole non-linked thing, if we're thinking non-linked, we should be picturing separate autosomes for each trait. And when we think about things being diploid, we should picture backup copies of those homologous chromosomes. And again, in this generation, they're all the same. So again, I set this up so that we can kind of picture what Punnett squares are all about. It's easy to get the letters down and figure all that out, but from a meiosis standpoint or from a chromosome standpoint, it's important that we kind of know what's going on. So with meiosis, the gametes are haploid. And again, these are the full diploid parents. And then the idea behind a Punnett square, inside the boxes would be the next generation and all of the possibilities of offspring. What we put around the outside are all of the possible gametes. And with them being haploid, a gamete only contains one copy of each autosome. So when we start thinking about this, what would be one possible sperm that could be produced? It could get this G and this D, and it would be haploid. See, here's 4 letters, here's 2 letters, and here are 4 chromosomes, and here are 2 chromosomes. Again, we could go this G, and it's not going to be GG, it has to be one of these and one of these, because you need every gene in a gamete, but you're only going to have one version of that. So again, it could be this one and this one, it could be this one and this one. That's what I'm showing there. Next one, it could be this G and this D, or it could be this G and that D for the last one. We do the same thing on this other side. Could be, like if I were to number these, 1, 2, 3, 4. It could be 1 and 3, 1 and 4, 2 and 3, and 2 and 4. And the thing with this, because they are homozygous, every single possible gamete is the same. So P1 and P1 makes F1. And F1 is going to be all of this. And from before we talked, F1 is all heterozygous. So F1, we take the gametes, and each box represents a possible zygote or a possible fertilized offspring. So we could take that, and when we fill this out, we take this G, match it up with that G. We take that D and that D. And again, what we're going to try and do is put, if they are heterozygous, we put the dominant one before the recessive one. So fertilization now, as you can see, it brings us back to diploid. We've got backup versions or diploid. We go to haploid, halves, and fertilization brings us back to diploid. And that's one way to figure out if you're doing this right. If you have 4 things here, you got to have 4 things here. So again, GG, DD, GG, DD. And then as we keep going here, because all of these are the same and all of these are the same, we can start skipping steps. And again, the F1 generation here is all heterozygous. So if we were talking about what are the phenotypes of this F1 generation, we've got GGDD. And this one right here was a green dwarf. This one was purple tall. But every single one of these, from a phenotype standpoint, are going to be green dwarfs. There will be no purple talls in the F1 generation. But all of them are carriers because they are carrying the allele for purple and carrying the allele for tall. Okay, so the question then, this first part, again, this was the setup. It gives you this picture. It says this is chromosome 1, this is chromosome 2. It talks about having F1 progeny, and it says let's first get this F1 progeny thing settled. So again, the P generation, it's saying we've got a GGDD, which would be that, crossed with GGDD, and we will produce F1 progeny, which is that. Okay, again, heterozygous. This is F1. And if we're to take this idea of F1 and stick it into their diagram here, we would have it look like that. Now again, you kind of see these two things, and that could be a little bit confusing because you might be like, oh, this one is the big G and this one's the little g. But in this setup, the way they've drawn this, there's diploid number of chromosome is 4, and this is 4. They don't have 4 here because what they're showing is already past the S phase of interphase where sister chromatids have been produced. So this is the big G allele, and you have a sister chromatid of it as you start getting into meiosis. So this is what you should be picturing according to their diagram, because you have the sister chromatid right next to it. So when we talk about the homologous chromosomes, these are both homologous to one another. This is the backup to that. However, it's already made a sister chromatid and already had that replication of DNA. So it gives you these 4 circles here. It says construct a diagram to depict the 4 possible normal products of meiosis that would be produced by the F1 progeny. Again, they use all these big fancy words. Basically, if you have this, what are the gametes going to look like? What are the products of meiosis? What we're saying here is what are the sperm or egg actually going to look like on a chromosome standpoint, and then sticking letters with that too. It says show the chromosomes and the alleles they carry. And when we talk about the alleles they carry, they're talking about which letters are you going to stick on there. So assume the genes are located on different chromosomes. And again, that's that non-linked part, which is this is why we're picturing these on separate autosomes, because they're non-linked. We'll get to linked in a second. And so you're supposed to, the flower color is on chromosome 1. So that's why I put the G's on these ones here. So F1 gametes. What possible combinations can we have here? If we go back to that idea of 1, 2, 3, 4, 1 and 3, 1 and 4, 2 and 3, 2 and 4, that'll give us our different possible combinations that will be haploid. So we're diploid here. We need one of these and one of these haploid. So we could do that, 1 and 3, 1 and 4, 2 and 3, and 2 and 4. Again, these are all haploid. They contain all of the genes, but only one of the alleles. Okay, so now this is my little chromosome version of this. Based off of this, what would it look like down here? And it might look like that. Okay, so again, same kind of deal. They want you to show the chromosomes and label the alleles they carry. Again, big G, big D, little g, little d, they don't have to be in this order or my order, but as long as you have all of the different possible combinations from the heterozygous generation, that's good to go. Okay, so we're going to take this one step further before we head over to understanding linked genes. So what about this F2? Because all of these are green dwarf, right? But they're carrying these alleles for purple tall. And when we cross up two heterozygous individuals, we just looked at the combinations of gametes that could be produced. But what happens when you cross them all up? What do we end up with in the F2 generation? So again, this is an F1 cross. We have heterozygous, looks like that. We've got heterozygous, looks like that. And again, we're thinking about the different possible gametes, which we just did in that last little thing there. And 1, 3, 1, 4, 2, 3, 2, 4. So we could have that G and that D, that G, that D, that G, that D, that G, and that D. And that's what we just did. Now, again, the other side would be basically the exact same thing. And now, as we go through this, we need to make the F2 generation. And a big part of this is like, what is the probability of how many of each type of phenotype are you going to get in that next generation? So when we're doing dihybrid crosses, ultimately there is a pattern to all of this. So we start matching up the letters again. G goes in G, D goes with D. Again, we're crossing this and this to make that box. So it looks like that, G and G, those D and D. Those G's, those D's, G, G, D, D, and you keep going. So again, before I hit the button, as we go through this little tutorial here, it's important not just to watch, but try and anticipate what things are going to be. So this next box, try and pick out what it should be. Does it look like that? Next one, what should it be? That. All right, hopefully you're getting these, matching things up, how they should be. All right, so now we got a whole bunch of letters here, don't we? But what's the whole point of this? So a lot of times we want to understand the F2 phenotypes. What physical characteristics is that F2 generation? Again, this is all F2. These were F1 crossed with another F1. This is all F2 progeny. Okay, so F2 phenotypes. What different possible combinations can we have? Now again, understanding what is going to show up, anytime it's heterozygous, the dominant will show up. The only way we get the recessive allele in the phenotype is if it is homozygous. So we could have a green dwarf, we could have green tall, we could have purple dwarf, we could have purple tall. And when we change these over to colors, again, this would be green dwarf because it has big G, big D. This would also be green dwarf because although it's heterozygous, the big G and the big D is what you would see in the phenotype. Same thing with this one. This is just like the parent. Again, we go through this. Now, what is green tall? Green tall, again, could be GG or big G, little g, and it would still be green. To be tall, though, it has to be homozygous recessive. So that's a yellow, or a green tall, sorry. That's a green tall, and that's a green tall. Purple dwarf, it has to be little g, and then it could be big D, or big D, little d. And these three match that. And this one right here, purple tall, is little g, little d. So when our F2 generation will always follow a 9-3-3-1 pattern. It will, so there's 16 possible outcomes here. 9 of them will be a particular dominant phenotype, and then it'll be 3, 3, and 1. We'll only have one out of the 16 that demonstrates phenotypes are both recessive alleles. Okay, so now the next part of that question, after we drew the different possible gametes and what they would look like with which letters, is to predict the possible phenotypes and their ratios, which is what we just did for the F2 generation. But instead of F2, they're doing a test cross between F1 and a homozygous recessive. So we are doing an F1, which is heterozygous. And again, it doesn't tell you what the letters are for F1. You need to know that F1 is heterozygous, and then GGDD. So GGDD is a purple tall. Again, we're test crossing non-linked. We've got that heterozygous and that homozygous. Again, 1, 3, 1, 4, 2, 3, 2, 4. And then these are all the same, right? Because you can have this one and that one, this one and that one, this one. They're all GD. They're all little g, little d. So really what we're most concerned with is only this top row right here, because all of these are different. And again, this first box is filled out. We put the big G with the little g, the big D with the little d. Next one, big G, little d, little d. Next one, and next one. Okay, so it said what would be the possible phenotypes and the ratio. So because all of these are the same, once we have the first row done, it basically copies all the way down with all the rest. It just follows that same pattern. So again, what are the phenotypes? That's green dwarf, that's green tall, that's purple dwarf, and that is purple tall. So based off of this test cross of an F1 and purple tall, we end up with 4 possible phenotypes, and the ratio is a 1 to 1 to 1 to 1. And again, any test cross that is non-linked dihybrid with F1 and the homozygous recessive for both alleles will always give you a 1 to 1 to 1 to 1. Okay, now we're getting into this world of linked traits. And the question is, if two genes were genetically linked, describe how the proportions of phenotypes of the resulting offspring would most likely differ from those of the test cross between an F1 individual and a GGDD, a homozygous recessive individual. So now we're getting into this whole linked business. I'm going to take you through a P1 cross, and so that you get this idea of how linked traits differ from non-linked traits. When we're talking non-linked, again, it's talking that the traits are on separate autosomes. If they are linked, the traits are linked on the same autosome. So even though you have GGDD and you have 4 letters, you only have 2 chromosomes. Whereas this, everyone represents. So this actually almost gets back to a monohybrid cross. It's just with multiple letters. So let's take a look at what that looks like. A linked P1 cross. We've got this P1. Now, with that linked idea, what can you picture in your head from a chromosome standpoint what that looks like? It is still diploid, but because it's linked, they are on the same chromosomes, and you don't have 2 sets of autosomes even though you have 2 traits. You still have a backup version of every gene because it's diploid. This other P1, again, they're linked, they're recessive because when we're talking P1, we're talking true breeding, and true breeding, which means they're not carrying any different alleles than what they are displaying. So haploid gametes, how do we do this? What is a possible gamete here? So we're not doing 1, 3, 1, 4, 2, 3, 2, 4. What are the different possible combinations? And I'm a little misleading here because I'm showing this big box. And really, it gets back to the same old monohybrid. The offspring can either be this or this. Same thing with that other side. And then we just do it like the old-fashioned way. And you match those up. This, you'd have that in that F1 generation right here. You'd have this fertilization here, and it would go back to diploid. Again, it's still the same amount of letters as this. It's the total same number of chromosomes as the parents. And now we're looking at this, even though these are still linked on the same chromosome, you would still write out the genotype for this F1 generation as big G, big D, little d. And again, because all of these are the same, it's all, this whole F1 generation is identical from a genotype and phenotype standpoint. They're all heterozygous and display the dominant phenotype. So the F1 genotype, heterozygous, phenotype, dominant. Okay, so now let's get actually into the question a little bit here. Predict the possible phenotypes and their ratios in the offspring of a test cross between an F1 individual and a GGDD individual. And that's where we were 1 to 1 to 1 to 1 with this one, with non-linked. Okay, now it's saying, how would it be different? How would the proportions of phenotypes differ from those of the test cross if they were linked? So if this is how it is, how would the ratio of phenotypes in the offspring be different here compared to here? So let's line these up and think about this. And again, I want you guys to, in your mind, picture what this should be. So again, the P1 generation, they're all true breeding. And what are those chromosomes going to look like if it's linked versus non-linked? Non-linked would look like that, and linked would look like that. Okay, so we cross up our F1 generation. F1 is heterozygous, non-linked, like that, linked, like that. This is still heterozygous. This is the same genotype. However, when they're linked, this does not look like this. And therefore, in this test cross, we get different varieties. This one, again, because this right here is going to have the same gamete for every single one down, I skip it and just do the top row. So when we're thinking F1, what are we going to have up here as far as possible gametes? This is the 1, 3, 1, 4, 2, 3, 2, 4. And then we're going to have that gamete from the true breeding homozygous recessive, which gives us those 4 varieties of phenotypes. And now on the bottom here, I'm playing out this. Okay, it is 1 to 1 to 1 to 1. Green dwarf, green tall, purple dwarf, purple tall, and you'd end up approximately 1 to 1 to 1 to 1 for offspring, if it were 100. Now, this one, again, when they're linked, it's more like a monohybrid cross instead of a dihybrid cross. So if we're thinking what kind of gametes are going to be produced from this F1, it would be that one and that one because they just split apart. And then over here, we've got that homozygous recessive. We end up with, when they're linked, they end up looking like their parents. Okay, so right here, the parent was here, parent was here. When they're linked, the offspring look like the parents, whereas the offspring of this cross, when they're non-linked, you start bringing back in some of those recessive alleles. So, if we're taking a look at this and we had 100 offspring, what would be the ratio? Okay, now what in the heck is going on here? Because this is 50-50, right? 1 to 1. And now my answer here includes some of these ones. And where in the world do these come from? So let's figure that out. Anybody have any guesses whatsoever? It is from crossing over. Holy smokes. So non-linked is just getting a little bit more confusing here. So when we cross over, we can get recombinants. So in this tetrad formation, when the homologous chromosomes come together, again, you have your sister chromatids here. And when we have this tetrad formation, we can swap pieces of chromosomes. And then as we divide, we can have whole new combinations. And that's where you get in some of those old ones. So again, based off of my little golden chromosomes here, let's take a look. You can have this or this. They're linked, right? But now don't forget, we've got sister chromatids. And those sister chromatids in the process of meiosis, during meiosis 1, at metaphase, the homologous chromosomes form tetrads. And in that tetrad formation, you can have swapping of crossing over of pieces of chromosomes. So here you can see that as these are all wound together before we have our first anaphase, we can actually slip off and swap these back, which then ultimately, instead of this being GD and GD, instead of these going straight down and doing those 4 possible combinations here, we could get this combination, which is more similar to non-linked traits. So that's where we get in those green dwarfs and purple talls. That's where they come from because of crossing over. And again, most of them are still going to be looking like the parents, but we could throw in a couple of recombinant alleles. One last thing to think about here, if those linked traits are very close to one another, you are going to get fewer recombinants because as they cross over, if they're very close together here, if they're both on this bottom part, when this swaps with that, it still stays little g, little d, and this one would still stay big G, big D because they're swapping together. In this one, it was swapping one of the traits, but not both traits because they were far apart on the chromosome. The closer they are, the more linked they are, and the fewer recombinant alleles will exist. So linked, non-linked, tying that all into Punnett squares and tying into this world of meiosis and gametes and diploid and haploid, and that is the end.